Assalamu alaikum everyone. I am Asifar Rahman. I graduated from Noakhali Science and Technology University in Biotechnology and Genetic Engineering. In today, I will talk about PCR, which is called polymerase chain reaction. At first, we need to understand why PCR, why this is so significant. Suppose a crime has happened and the police actually got some evidence like some biofluids. This could be semen, blood, or urine sample. From this small quantity of a sample, I only got only tiny bit of DNA. So it will be very difficult for me to extensive analysis and storage of this DNA. So I actually need to amplify my sample DNA. So for this reason, this technique actually very significant in life science. So the thing happens is that suppose this is sample DNA. This sample DNA give rises to two sample DNA. This two sample DNA further give rises to four sample DNA. We can see that the exponential amplification of our sample DNA. So if we can amplify our sample DNA, it will be helpful for us to further analysis of that sample DNA. So now I will talk about the principle of PCR. So the principle of PCR actually depends on three steps, which are denaturation, annealing, and extension. First, I will talk about the denaturation step. So, what happens here? Suppose this is my sample DNA, which is a double stranded DNA. Now, if I apply high temperature, which is almost the boiling point, my single stranded DNA will be double stranded. So this happens in denaturation step. Then what happens? The next step is called annealing. Annealing of what? Annealing of primer. We know that to extend or to synthesize the nascent DNA, the DNA polymerase actually needs primer. So we know that the template DNA is in 3' to 5' direction. So one primer will sit here and another primer will sit here. So annealing of our primer is done. This will be called our forward primer and this will be called our reverse primer. Then so another thing is that the annealing temperature is almost 55 to 56 degrees Celsius. Then the next step comes, which is extension. So our in this step, the DNA polymerase sits on the primer and he adds the nucleotides and synthesizes the nascent DNA. So the temperature for expansion is about 72 degrees Celsius. So we see that from a single stranded DNA we actually got a single double stranded DNA, we actually got two double stranded DNA. So this is the basically the principle of PCR. The interesting thing is that this polymerase enzyme is not actually this DNA polymerase enzyme actually special DNA polymerase enzyme. This is not a regular DNA polymerase that is found in human body. So this DNA polymerase actually called Taq polymerase. Taq polymerase. T comes from the name thermos. AQ comes from the name aquaticus. So Thermus aquaticus is a bacteria which is found in hot spring. So this bacteria can actually survive in high temperature. So the DNA polymerase from this bacteria can persist the high temperature which I apply to denature the DNA, which is actually 95 to 98 degrees Celsius. So instead of using regular human DNA polymerase, we actually use the DNA polymerase which is found in this bacteria. So thank you everyone. This is all for today. Hopefully I will give another insight about PCR in the next video.